Stanford University.
My name is Greg McAllister. I'm a computational biologist at the Novartis Institute for Biomedical Research. I'm going to talk a little bit about drug discovery in the era of big data. As you might imagine, big data means something a little bit different in the pharmaceutical world. We traditionally don't have lots of log files that we need to parse or large social networks we need to analyze. Our principal problems are increasing number of technology used to investigate the biological domain that are producing larger and larger data sets. So our challenge is to store, process, and analyze this data to try to come up with some sort of meaningful biological conclusions. So given that Novartis doesn't generally present at conferences such as XLDB, I thought maybe I'd just give a quick background of sort of who we are and what we do. So NIBR is the research arm of Novartis Pharma. We have a unique research strategy driven by patient needs, where we try to identify unmet medical needs and sort of gain more insight into the mechanistic understanding of this disease, with the idea being that those insights would transfer across diseases to larger markets. We're about 5,000 scientists globally. We're headquartered in Cambridge, Mass. We have increasing focus on molecular pathways that are shared across various diseases. Again, with the idea being that elucidation of function of these pathways can be beneficial across various disease states. We progress potential therapeutics from early target discovery through first phase clinical, which allows us to gain clinical insights into understanding these diseases. Given that we're based in Cambridge, which is a large academic and biotech environment, we're keen to form strategic alliances with both these populations, sort of help us bridge any technological gaps we may have. So what do we do? Aside from our general biological experimentation and exploration, we focus on multifaceted set of core technologies. Aside from small molecules, we're interested in antibodies, siRNAs, and protein therapeutics. We have a strong focus on medicinal chemistry, where we try to optimize small molecules for increased efficacy against targets. The world that I'm in is target discovery pathways, where we're interested in the interactions of genes and proteins within a cell, the structure of networks and their effect on disease. Assay development, where we generate high throughput screens where we can test identify the target of millions of compounds at a time. Preclinical safety, where we assess the toxicity of potential compounds, as toxicity is the #1 reason why drugs fall out of the clinic. Translational medicine, where we go from early stage drug discovery into the clinic, into humans. So how do we do it? Principally, traditionally, drug discovery has been sort of a one target per disease approach, where we try to identify some sort of therapeutic that modifies a target with the hope that it has some sort of effect on the disease state. Most of you are probably aware of the sequencing of the genome late 90s, early 2000s. This sort of gave us a compendium of all the potential all the genes and targets that are potential genes and proteins that are potential targets. The idea being that this would increase the number of drugs that are actually being developed. Of course, this hasn't happened. So the approach we are taking within DMP is to focus on the interaction of these proteins within a cell. So proteins form protein networks, molecular pathways, signaling cascades that are functional units of the cells. So we're attempting to elucidate the structure of these pathways, the interaction between these pathways, and the effective modulation of these pathways on human disease. There we go. So given we're interested in pathway analysis for target identification and validation, how do we go about doing it? So we have multiple orthogonal approaches into pathway elucidation. Genomics being one of them, where we're looking at the activity of the entire genome within a disease state or cell line at one time. So we're interested in transcriptional regulation of genes, and the effects of perturbation on the genomes. Proteomics, where we're looking in the protein space, we're interested in identifying the potential therapeutics interaction with proteins interaction with each other. Model organisms, We model disease states within animals, with the idea being that we then can test perturbation of certain pathways and its effect on disease. This gives us an in vivo approach as opposed to in vitro. High throughput screening, again, where we can test millions of compounds, identify their targets through assays. Computational biology, which is the world that I'm in, which is sort of making sense of all this data, putting it together, coming up with biological conclusions. RNAi technology, where we can silence, knock out individual genes and understand its effect on the signaling cascade and a pathway, biology in general. So given all these things that we work on, what are the hurdles that we're currently facing, at least in the world of computational biology that I'm in? So as we've talked about a lot here this week, we've increased in data size. Our data size at the moment is primarily being driven by next generation sequencing, which you just heard about. So this is a figure from a paper that was recently published that just gives the rate of sequencing increase in comparison to Moore's law and Kreider's law. Basic idea being that we're sequencing base pairs faster than we can actually analyze and store it. Increasing dimensionality of data. So within NIBR and data that we bring in from the public space, we have multiple technologies that are providing orthogonal views onto a biological space, and sort of understanding and being able to analyze these data sets together is increasingly complicated. And then put these in the context of a biological organization, the signaling cascades and the pathways that make up a cell. It's becoming increasingly harder to do these sort of analytics. So analytics to NIBR, where are we? Sort of what we need. So NIBR is a pretty big organization, so there's a lot of people that do a lot of different things. So you name it, someone in Novartis probably does it. But we have your general standard solutions for analytics, for statistics. R, MATLAB, S, your programming language of choice. For HPC type applications, we have Linux clusters. I don't remember how many nodes there are. They're pretty big, probably not compared to what some of you people here deal with. But we have people that write custom MPI codes for particular algorithms that are slow and need to be sped up, that need to work on different data sets. It's a pretty limited pool within Novartis, and as you all know, it takes a while to write MPI code. Traditionally, we have all your normal databases, the Oracle, MySQL, Postgres. This is generally where we store our raw data. And of course, the analytics that we can do within these databases, it's somewhat limited. We've been exploring the last year and a half or so, moving some of this analysis and data storage to Hadoop and Mahout, sort of a work in progress. But our principal problem is that these data sizes are growing rapidly. We don't exactly know where they're going to stop. We don't know what technology are going to come down the road. So our principal problem lies not in, well, it lies in storage, but our principal problem lies in the analytics over these data sizes. Like I said, we focus mainly, at least in our group, we focus mainly in R. So we're limited to in-memory type applications, embarrassingly parallel type applications. Where we really want to be, sort of our ideal world, probably a lot of people's ideal world, we want to develop some sort of system or have some sort of system that mimics R, that works on terabyte-sized data sets. We need a sort of a large-scale analytic system that just works, so to speak. Multiple users from all sorts of backgrounds, certainly not comp sci backgrounds, certainly not development backgrounds. Scientists, biologists, analysts, developers with a broad range of skill sets that don't necessarily have the ability to write MPI code or even work with clusters. So ideally, we would have some sort of R system where you would type all your normal R functions, and it would just work no matter what the data size is. I realize this is sort of ideal, and this is what everyone wants, but this is sort of the state we're in. So about a year ago, we started a collaboration with CyDB and Paradigm 4 people to investigate whether CyDB could be used to this end. So how far we could take CyDB and our analytics needs. So most of this is probably well known to this community. CyDB is a data management and analytics system. I believe that's the phrase they're using. All open source, and then Paradigm 4 built on top of this. The principal data model is nested multi-dimensional arrays. In the end, most of our data types coming from biological space, they're still just matrices. We end up with just matrices of compound gene effects, big matrices, multidimensional. Array cells can be tuples of values or other arrays, so we can have nested arrays. Arrays can be sparse. This is big in our world because We can generally have screens that will go against a certain number of compounds, but we may screen our full deck of 1 million compounds, or we may screen partial of it. So we end up with sparse matrices, just by nature of oftentimes it's too expensive to run everything against everything. Data storage. So arrays are chunked in multiple dimensions in storage. Chunks are partitioned across multiple nodes. Each node is shared nothing. Chunks have adjustable overlap, which of course becomes important depending on the algorithm you're actually trying to implement, particularly in image analysis type of applications. Architecture, shared nothing cluster, which is big for us. Novartis is fine buying lots of commodity boxes, not so good at $1 million, multi-million dollar storage systems. Queries refer to arrays as primary data types. Again, it's the abstraction of the sort of array chunking multiple dimension storage system into something that just says, give me the correlation of array B, that sort of thing, something that's easy. Query planner optimizes specific functions. So this is more of the SciDB development team, but the ability to sort of analyze or access different parts of the matrix depending on their algorithm you're actually running. So I just wanted to highlight a couple features of the SciDB model and why it's important to us, why we started thinking about it. And again, most of our data types end up being matrices and arrays. And we end up with, again, orthogonal views onto biological space that end up being sort of multi-dimensional arrays. So these are examples of just profiling technologies that we have within NIBR, where we're taking large collection of compounds, screening against biological assays, and ended up with some sort of profile of some dimension of biological space. So we have this promoter signature profiling where we can ask what pathways are being perturbed by a modulator. We have LMF, which is a transcriptional readout, tells us what genes are modified transcriptionally upon compound perturbation. HipHop is a yeast deletion screen where we can identify very clearly what the target of the modulator is in yeast. In chemical proteomics, where we can answer what proteins bind to my compound. And so one important thing here is that these are actually different levels of throughput. So chemical proteomics is a very low throughput, whereas PSP and LMF are very high throughput. So we have, this is sort of go back to the sparse arrays of, we end up with these large-scale multi-dimensional arrays where good portions of it may be empty just by nature of the fact that you can only run so many compounds through chemical proteomics. So natural extensions to this, right? So simple things, subsetting by different dimensions, which at the moment is hard. So each one of these is in different database stores at the moment. We can ask simple questions like correlation by each dimension, which isn't that simple once the data gets big. But and then we can ask questions across these sort of correlation matrices of where they intersect. So what part of these different profiling platforms are telling us the same thing about biology? What part of them are telling us different things? So it allows us to do sort of a gap analysis type of thing on biology. Multidimensional statistics. These are the kind of things that we'd like to do. I'm not a statistician. I don't know exactly what that is or what that would look like, but that's sort of where we want to go. Uncertainty. So SciDB is building in the concept of uncertainty, principal normal distribution type uncertainty, standard deviation, standard errors. So this is a big thing in biology. Everything we do is experimental data. We run everything in replicate, so we get errors, right? So on the left-hand side, we actually have a, I don't remember what that's actually from, but it's a compound screen. Against an assay, we run multiple concentrations of a compound, and we generate an IC50 of inhibitory concentration where there's a maximal 50% inhibition across some sort of target. But basically, we're curve fitting data points, and we have an error. And what we end up with is a single IC50. But often, once we take this IC50 into downstream analytics, we lose the ability to sort of propagate this error. On the right-hand side, we have a transcriptional readout where we're looking at 100 different genes in response to a compound perturbation. And these are run usually in triplicate, 4 doses or whatever it is. And we get some sort of error bars. And the general analysis that happens here is we take these profiles and we look for similarities and correlations to other compounds. So of course, as soon as you do that, you lose the concept of error bars. So this gene, of course, has a huge error right here. So we might be a little skeptical about how reproducible that is. But once we start doing things like correlations, we lose that. So it's of interest to us to be able to take these experimental measures that have this inherent error and propagate them through various analytics. And again, It's a question for statisticians. The propagation of error, I don't exactly know how that works, somewhat complicated. But these are things that we want to be thinking about. And at least at the raw data level, be storing it and be aware that it's there. Data persistence, provenance. So this is a big deal in our world. The ability to generate results and then backtrack to figure out where that result came from. Our data sets change frequently. Depending on the data size, we don't necessarily store all those changes. It's a problem we have. So CyDB builds this sort of data persistence provenance into the database system. So arrays that are created are immutable unless specifically defined otherwise. So we can do things like create updatable arrays where we can actually change them. But even when declared as updatable, original cell values are never actually overwritten. So there's a new version created. So we can always go back sort of in time, right, and redo an analysis that's been created depending on the date that it was generated. So log files generate repeatability of data derivation. So we have the ability to go back in log files and look and see what happens. So it's important to point out that these are all things in CyDB that are not necessarily production, being worked on, being developed. So I can't necessarily do all of this now, but this is sort of what we need to do where the CyDB and Paradigm 4 team is going. So we get a lot of hassle within Novartis for keeping some sort of lab notebook, which we're basically, computer scientists. We're not very good at keeping lab notebooks and write everything down. It seems a little bit opposed to everything we do. So this sort of system would build some sort of a lab notebook for us. So an example use case of where we're starting with using CyDB, an example data set, if you will. So we have a large compendium of microarray data. Microarrays are just platforms for simultaneously measuring the expression levels of thousands of genes. So we can treat various cell lines either in normal quiescent state or the result of some sort of compound perturbation and measure the expression levels of thousands of genes at once. So this technology has been around for quite a while. It's fairly well studied within Novartis and certainly out. But so we've built a large database of approximately 6,000 of these arrays. Each array being about 6,000 probes, probe representing theoretically, hopefully, one gene. It's greater than 100 indications, indication being disease state, primarily sort of oncology, but different disease states or normal states. So data matrix results in about 6 billion data points. At the moment, we store it all in a Vertica database, and this is sort of our method of extracting and querying the data. And we do simple analytics, sort of the standard deviations and z-scores and these sort of normal Vertica type analytics. This data set has multiple use cases. Target finding, as I've sort of explained. Patient stratification, where we can identify patients that might be more susceptible or better suited for certain therapeutic intervention. And biomarker identification. So the end result is we have a large matrix of samples, sample being a sort of a disease state or a normal state, probes being a gene, and metric being some sort of normalization. So as with any data sets, there's multiple ways to sort of normalize it and look at it. And depending on the application or the need, we process it in different ways. So we end up with sort of this three-dimensional data set. So it's important to point that this is sort of where we are now. We've built this big microarray database. It's big, it's whirly, it's hard for us to analyze. But it's quickly being replaced by next generation sequencing, which in essence generates the same sort of data type. However, it's a digital readout. So we've talked about this a little bit more this conference, so I don't want to repeat too much, but NGS is highly reproducible, highly sensitive. It detects about 25% more genes than a microarray. Microarrays are designed, so you design probes for certain genes, so you can only measure what you're actually designing for. Whereas next-gen sequencing can theoretically measure anything that's actually out there. In general, it's 2,000 times more data points compared to a microarray, ballpark. As far as data sizes, I mean, we've talked about this, but it's basically a gigabyte per lane, terabyte per experiment. Once you scale up to the size of the experiments that we already have in Disease Express, it's the petabyte scale. So we sort of came up with this sort of analogy when we try to explain the problems we're running into to sort of our upper management, if you will. Microarray, what we have today is the equivalent of sort of storing and mining the names of all the people in Eurasia. And so when we move into NGS and sort of duplicate this microarray data set in NGS, we're dealing more along the lines of storing and mining the names of all the people ever born in the history of the world times 90. So I don't really need to explain data sizes to the people in this room, but this is sort of the scope of what we're dealing with. So I wanted to give you sort of an overview, this being a technical crowd, sort of what it actually looks like. So we've loaded all this data into SciDB and started sort of looking at it, analyzing, and seeing what we can do, seeing how far we can take SciDB right now. So SciDB has two interfaces to actually query the language, actually query the database. A SQL-like syntax, which is extremely important for a group such as ours, because SQL is established, people know SQL, they feel comfortable with SQL, so you sort of have to have that interface. And then a functional language, which is, for people like myself in the computational biology world, we're used to R, we're used to functional programming languages. This feels a little bit more comfortable. I feel like I can sort of express what I need to do a little bit better. So this would be an example of just, we're actually loading array express into SciDB and doing a simple Pearson correlation, which I'll go into in a second. And also extensibility. It's hard for database systems to have every single mathematical function or probability distribution or whatever it is. So we need to have the ability sort of to extend SciDB. And importantly, again, have the UDS operate on arrays. So not have to deal with sort of chunking and data storage and where things are located. So just a couple of example use cases with timing, where we are with where the SciDB team is right now. So this would be a simple, just create an array of response values. I don't remember what empty indicator is. You have to ask Paul what that means. Basically, of 3 dimensions: algorithm, sample, probes. So we're ballpark, 65 by 50,000 by 24. So if we start doing simple queries, this is intended to be sort of a database management system. So we need to ask basic queries, pull basic data stats out, aside from doing actual complex analytics. So if we do simple things like count the elements of one slice of array. So if we pull, we slice the array by the 2 big dimensions, so 60 by 50,000 for a particular type of algorithm, and just count how many there are. So we're in the ballpark time of 10 seconds approximately. Find the min-max of an array across 1 dimension. Again, min-max across the 60 by 50,000. So as you can see, 41 minutes, not exactly what you want to write home about, but point B is to the point B is where you can actually do this. And these times are going to go down considerably when the Paradigm 4 SciDB team starts focusing more on time. Start asking questions like average response across the sample. So again, we're slicing the dimension, slicing the array in the large dimension, looking for average response times. We're down around the minute timeline for these sort of queries. So a little bit more interesting, we're going to start asking larger scale analytic questions, right? So basic questions you want to ask of this sort of data type are, what genes have similar expression profiles? What diseases look like each other? We're interested in finding pathways that are identical across disease states, these sort of questions. And so there's an abundance of literature that's done this on a small scale. People have worked with microarrays for a long time. But we want to start doing things on bigger data sets, seeing if we can do things on bigger data sets, seeing what the results start looking like. And there are all sorts of caveats to this, but this is the general exploration that we want to do. So sort of as a first test for SciDB, we want to calculate the Pearson correlation across the whole database. So your data matrix is approximately, again, 60,000 by 60,000. Right now we have this set up on 4 nodes, dedicated hardware, 16 cores, 2.4 gigs, terabyte drives, so single drives. And so at the moment, again, this is a build as of now, the release as of October 2011. It takes about 270 minutes to do this entire Pearson correlation calculation. So it's important to note that, first of all, this time will inevitably get faster, but it's still pretty good. I mean, we can't actually do this, at least in the hardware we have right now, we can't actually do this calculation. So this is the first time we actually generate this entire matrix. And this is actually the query that generates the Pearson correlation matrix, where we're basically slicing the matrix twice. So we're looking only at one algorithm, one different type of normalization, and doing the Pearson call and storing it back into the database. So you see we have this sort of functional programming language that allows you to easily deal with these sort of analytics on big data. So another thing we were interested in was biclustering. So traditionally, we do things like the correlation. We generate some sort of distant matrix. We cluster. We start seeing what things are like each other. Another approach is to do a simultaneous biclustering, or biclustering by rows and columns at the same time, as opposed to everything sort of all at once. Again, this is the idea here is that we sort of elucidate certain parts of gene expression space that correspond to pathways that are implicated in multiple or overlapping diseases. So biclustering's been around for a while. It started to get applied in the bioinformatics realm 5-6 years ago, I guess. But we're interested in sort of questions of combinations of gene samples that correlate strongly with each other. So just last year, there was a paper that was published that uses SVD to do biclustering. So we asked the Paradigm 4 team to see if they can implement this inside EB and what it would look like. Because again, we can't run this algorithm on this data set with the machines we have. It's just, it runs out of memory every time. So they developed this sort of hybrid R side EB algorithm. So right now it's only been done on the 60,000 by 2,000 ballpark matrix. So it hasn't been done on the full matrix size yet. SVD is a little bit computationally difficult. So they run on EC2s, again, so 4 nodes, 2 6 gigahertz, 8 gigabytes of RAM. So it's an iterative algorithm where you're sort of trying to minimize an error. So doing it on 3 iterations takes about 31 hours. So it's about 10 hours per sort of SVD on these big data sets. And again, this is the SciDB October 2011 build. But again, this is sort of building to a place where we can do these sort of analytics on data sizes that we haven't been able to do before. There's the last slide. So where does this all fit within NIBR? So, there's a lot of, you people all know far better than I do, there's significant momentum in the field of sort of big data analytics, different systems, the NoSQLs, all the things that are being developed right now. And so we follow all those, and sort of we struggle a little bit with how to sort of blend these new technologies with legacy systems. So we're dependent on the oracles and the MySQLs that store the raw data, and that's sort of what we have to work with. So we're really interested in just finding the right tool for the job. We have a pretty large Vertica store that we're building now to do bigger data sets. We're exploring the use of Hadoop and algorithms in Mahout. And it becomes a significant question of, if we're primarily interested in analytics, so where are we actually doing these analytics? What's the ETL part? What's the analytics, right? So you can do lots of things in Vertica. You can do lots of things in Mahout. Things Mahout can't do that sort of SciDB can do. So it's sort of a question of which one of these technologies is the best fit. So we're still working with that. Not sure exactly how everything fits together, but at least exploring all of these. I think that's it. That's it. So thank you.
Are there any questions?
In the 2 experiments you showed with both with 4 nodes, I'm just curious the scalability to hundreds of nodes. What does it look like?
I would love to be able to tell you that. I don't have the answer though. It's probably a better question for the SciDB people. We don't have 100 nodes to run at. I know they're working on expanding the EC2s to test the scalability. The reality is some of these Pearsons and SVDs, we really just finished recently, so the scaling part hasn't been done. I don't know if Paul or someone wants to add to that, but it's a good question.
Anyone else? Going wide? Okay, there's one.
Could you shed some light on how you integrated SciDB with R, with this hybrid solution mentioned?
Again, better question for the Paradigm people. So I think the basic process up until now, at least, was taking the pieces of this SSVD algorithm that were time-consuming and big, if you will, and pushing that down to SciDB. And I believe the interface right now is basically through the AFL, sort of the functional programming language. So you can execute those queries, much like you can in like an RSQL and RODBC. So you basically push that down into SciDB and bring the result back into R. Paul can correct me if I'm wrong, but so that's an important feature for us. I mean, how that interaction happens, and make sure you don't break R by bringing back huge data sets, these sort of things. It's where you sort of cross that line from the R interface and manipulation down to the SciDB part.
Okay. Thank you very much.
For more, please visit us at stanford.edu.